The second common type of substitution mechanism is known as the Sn1 reaction, in this case standing for substitution nucleophilic unimolecular. The way this is often observed is with bulky tertiary substrates. By way of an example, it's well known that bulky tertiary bromides, here we have T-butyl bromide, that these compounds don't react with good nucleophiles. Something like cyanide ion fails to react with this sort of substrate. But what's curious is they do react with what would be considered poorer nucleophiles. And that's shown here. This reaction happens quite readily. The T-butyl bromide is refluxed in methanol, and the product of that reaction is this methyl tert-butyl ether. The process is essentially reaction with solvent, and those sorts of reactions are known generically as solvolysis reactions. And so we're saying because the reaction is occurring with what is essentially the solvent, we simply call that a solvolysis reaction. It's known that this does not go through an Sn2 mechanism for a couple reasons. One is Sn2 reactions require a strong nucleophile. And we also know that they only happen with unhindered substrates because of the backside attack of the nucleophile. In this case, we have a weak nucleophile in the form of the methanol, and we have a hindered halide in the form of the T-butyl bromide. Another aspect of this that tells us it's not an Sn2 reaction is the kinetic experiments and what they tell us about the behavior. And when you do the kinetics experiments on this and vary the concentrations of these two substrates, you find that the rate of the reaction is only dependent on the concentration of the substrate. It is not dependent on the concentration of the nucleophile, as long as there is some nucleophile present. So this implies that the mechanism is operating often differently. And one of the things it implies is that the nucleophile isn't present in the transition state to whatever the rate determining step is in the reaction. It also follows that the nucleophile must react after the slow step in a much more rapid step in the process. So how does this reaction proceed? So here is the overall mechanism for this particular example of an Sn1 reaction. So the overall equation you see here, T-butyl bromide reacts with methanol under refluxing conditions to produce methyl tert-butyl ether. Byproduct of that reaction is HBr. What has been proposed and what's consistent with the experimental data is this. First, the carbon-bromine bond breaks, and this is the energy provided to do this is the heat applied when this reaction is refluxed. This is a reaction that is energetically uphill. Notice the equilibrium arrows here, that we are going uphill. The important thing is that some of this carbocation is produced. And this is a reasonably stable carbocation. It is a tertiary carbocation. The carbon that bears the positive charge is attached to 3 additional carbons. You'll notice that bromide is our leaving group. The next step, which is a much more rapid exothermic step, is that our nucleophile, in this case methanol, adds and forms a bond. So the carbocation acting as a Lewis acid, and the methanol acting as a Lewis base. This produces this oxonium ion, which can then be deprotonated by additional methanol to give our final product. And we are effectively producing HBr, but dissolved in methanol. So the hydrogen is in the form of this protonated methanol, and the bromine is in the form of the bromide ion. So much more complicated mechanism than the Sn2 in that we have 2 fundamental steps followed by a proton transfer. So we get ionization to form a carbocation, attack of nucleophile to form our bond to our new group, and in this particular case, because it's an alcohol, there's a deprotonation step that occurs also. If you map those 2 fundamental steps to the mechanism, the first 2 steps in the mechanism on the previous slide on a potential energy diagram with a reaction coordinate, then generically you see what we have here. And that is a rate determining slow initial step. So our substrate undergoes ionization to form a carbocation and a leaving group. You'll notice that the nucleophile here is unchanged. It does not participate in that step. So the transition state only involves the rupture of the carbon leaving group bond in our substrate. This produces a carbocation. It's a reactive intermediate. It's much higher in energy than where we started. And then the reaction that occurs is the nucleophile rapidly adds to that. This is a bond-forming reaction and thus has a low activation energy and is highly exothermic. And there's enough energy released that it drives the whole process. As you recall from general chemistry, the rate determining step is the highest energy hill that you climb in a reaction, and that is this step here. And so the overall process can go no faster than that initial formation of the carbocation, and that the overall rate of the reaction is going to be independent of the reaction with the nucleophile, that this step is kinetically invisible to the overall process. What's depicted here are just the two fundamental steps of an Sn1, ionization to form a cation, and then addition of a nucleophile. The particular example on the previous slide involved a methanol, so there was one small additional step, which was a proton transfer. So let's look at some of the details for an Sn1 reaction, and we're going to start with the effect of the structure of the substrate. And we find that the reactivity of substrates that contain leaving groups follows this pattern, that tertiary are more reactive than secondary, which are far more reactive than primary or methyl substrates. And then this follows the stability of carbocations that could be formed. The loss of a leaving group from a tertiary substrate would give a tertiary carbocation. Loss of a leaving group from a secondary substrate will give a secondary carbocation. As we've learned previously in the course, primary carbocations are so unstable as to not form, and methyl carbocations would be even less stable. Another thing we know is that substrates that can form resonance stabilized carbocations can quite readily undergo Sn1 substitution. This is an example here of allyl bromide. Allyl bromide can undergo ionization with a loss of the bromine to form a carbocation. At first glance, that carbocation appears to be primary, but it isn't fundamentally just a primary. It's a resonance stabilized carbocation, because we can, if we use our arrows to indicate how we draw resonance structures, we can see that that positive charge is delocalized over the two ends of the allyl unit. So this is a very stable carbocation, relatively speaking, and then nucleophile can interact with that. There are also substrates that generally do not undergo Sn1 reactions because they form very unstable carbocations, and they also don't undergo Sn2 reactions because for some reason the backside attack of the nucleophile is prevented. And those substrates are vinyl and aryl halides. Loss of a halogen from a double bond leaves a vinyl carbocation. It turns out they're quite unstable. Likewise, loss of a leaving group from a benzene ring leaves a phenyl carbocation. They're also very unstable. And then in addition, both of these substrates are somewhat hindered to the backside attack, particularly the phenyl one. The nucleophile would in effect have to be in the ring in order to interact with the sigma star orbital. And so we find that neither of these types of substrates undergo either type of substitution reaction that we've been talking about. So as I said, the structure of the substrate strongly affects the rate of Sn1 reactions, and it correlates with carbocation stability. And the more substituents you have on the carbon that bears the positive charge, the more stable that structure is. And we can actually calculate heats of formation for a variety of cations. And so you notice where the positively charged carbon is in these species. So the first entry here is a methyl cation. It has a very high endothermic heat of formation. It means it's energetically almost unobtainable. You can't make methyl carbocations. Primary carbocations don't exist in normal reactions because they're too high in energy. Getting to these numbers enthalpically is very difficult. When you get to secondary carbocations, you notice the carbon here that bears the positive charge has two carbons attached. Likewise here, this carbon with the positive charge has two carbons attached. Now you've dropped below 200 kilocalories per mole, and you're starting to get into the range where these can reasonably form in a chemical reaction. And finally, tertiary carbocations are the most stable of the alkyl carbocations, and those can form quite readily. So long story short, reactions generally occur either through tertiary or secondary carbocations, and we see no reaction for the substrates for primary and methyl substrates because those primary and methyl carbocations are too unstable. We also find in the Sn1 reaction a different stereochemical outcome than we do in the Sn2 because we have an intermediate formed. In the reaction by an Sn1 mechanism, the first step is loss of the leaving group to form a carbocation. Carbocations are a different hybridization than the substrates we started with. The substrates are sp3 hybridized and typically tetrahedral. Once you lose the leaving group and form the positively charged carbon, you end up with an sp2 hybridized species, which are trigonal planar and therefore achiral. The nucleophile then has an opportunity to add to that carbocation to either lobe of the empty p orbital. And that means that a chiral substrate will typically lead to a mixture of enantiomers as the final products. And you can imagine in a schematic sense here ending up with a racemic mixture. And we'll talk a little more about nuances of that here in a second. So looking at that from the point of view of a real compound undergoing a real reaction, here we go. Here is a chiral alkyl bromide. You'll notice that there are 4 different substituents on this carbon: bromine, methyl, this is an isopropyl group, and an ethyl. Loss of the bromine, or ionization of the substrate, produces the carbocation that is shown here. These are the 2 lobes of the p orbital. The colors are of no consequence. They simply represent the sign of the wave function. In this particular reaction, we're looking at solvolysis in ethanol. So the substrate is presumably heated in ethanol. Once the carbocation forms, ethanol can add to either face of the carbocation. It's a planar structure. And depending on which face that new carbon-oxygen bond forms, you get a different configuration at the newly formed chiral center. And you can see these are now enantiomeric forms. And you would end up, in principle, with a racemic product. While it looks like you would end up with a racemic product, it turns out that full racemization often doesn't occur. And there's actually a very good reason for this, but let's look at a couple examples of this and then see if we can understand why we're not getting exactly a 50-50 mixture. So in the first example, we have this tertiary alkyl chloride reacting with water. Acetone is just a co-solvent here. It doesn't participate. Water is our nucleophile. So we get ionization of this chloride and attack of water. After deprotonation, our products are alcohols. And you can see that one of those products has retained the configuration that we started with, and the other product has inverted. And these are the experimentally determined ratios. You can see that we've actually got less retention and a greater amount of inversion. The reaction has favored the inverted product. So not racemic, but a mixture of enantiomers, but a non-racemic mixture of enantiomers that favors the inverted configuration. It turns out that all of the examples of this sort of behavior always favor inversion. This example, the second example, is secondary alkyl chloride, which is also chiral in water, undergoes SN1, and we get 41% retention and 59% inversion. So why do we get a little bit more inversion than we do retention? It turns out that that observation arises from the formation of an ion pair when the carbocation forms. And that's illustrated here. So imagine what's happening in this reaction when this carbon-bromine bond breaks. And we form a bromide ion and a carbocation. Well, as soon as that bond breaks, we form a negatively charged ion and a positively charged ion. And as you are well aware, opposite charges attract. And so the simple fact is that when the bond breaks, the bromine moves slightly away, but it remains attracted to the carbocation now because of electrostatics. And so it tends to sit on the side of the carbocation where it broke off from. So in this particular structure, the bromine ends up sitting here on the top of this structure. That means it's going to impede the approach of a nucleophile. So water in principle, can add to either face of this carbocation, either lobe of the p orbital, but its approach from the top face of this structure is impeded by the presence of the bromide ion in this so-called ion pair. So the result is that in a collection of molecules, we get more water addition to the bottom face of this structure and less water addition to the top because of the impediment of the bromide ion. And the result is we get more we get more inversion of configuration and less retention of configuration because of the intermediate ion pair. For all intents and purposes, the ion pair is a chiral structure, and this is adding to the less hindered side. A few other aspects of the Sn1 reaction that are important are the nucleophile, the nature of the leaving group, and the solvent in which the reaction occurs. So first off, if you think about the nucleophile, the nature of the nucleophile actually doesn't affect the rate of an Sn1 reaction. Think about it. It's not in the rate law. It participates in a much faster step. So in general, we find that rates of Sn1 reactions are entirely dependent on the substrate that you use, not what nucleophile you're reacting with. The leaving group does affect the rate. It is involved in the rate determining step. It's involved in that bond breaking process. And so the more stable that leaving group is, then the more stable the products are that are formed. And so the faster the reaction. So it isn't just the structure of the carbocation that determines the overall rate of that slow step. It's also the structure of the leaving group. If this is a very stable anion, that's going to help promote the reaction. And then finally, solvents are important because we are producing ions, and those solvents need to interact with those ions. And so we find that polar protic solvents are the best because they can interact with both the cation and the carbocation, and they can interact with the leaving group. And these little arrows here are meant to represent solvent molecules that are very polar solvents. And we find that the negative end of these solvents are going to be clustered around the carbocations, and the positive ends of these solvent molecules are going to be clustered around the leaving group because it will typically be an anion. So we can now kind of compare Sn1 and Sn2 reactions. And if we just focus on the organic substrate that bears the leaving group now, and we can divide up our substrates into 4 categories. Tertiary substrates, where the leaving group is attached to a tertiary carbon. Secondary substrates, where the leaving group is attached to a secondary carbon. Primary substrates, where the leaving group is attached to a primary carbon. And methyl substrates, which are their own thing. And from the point of view of an Sn2 reaction, we find that the reactions are increasingly more efficient with less hindrance. And so Methyl compounds are some of the fastest to react under Sn2 conditions because the nucleophile can approach and interact with the sigma star orbital readily. The more alkyl groups attached to that carbon, the slower the reaction occurs. And very importantly, we do not see Sn2 reactions occur on tertiary substrates. For an Sn1 process, we find the opposite trend, that the efficiency of the Sn1 reaction increases as we go to greater substitution. So tertiary and secondary substrates react readily, tertiary more rapidly than secondary because they form more stable carbocations. We do not find Sn1 reactions occurring with primary or methyl substrates at all. Primary carbocations and certainly not methyl carbocations are viable intermediates in reactions. Finally, if we look one more time at Sn1 and Sn2 on different substrates, here if we focus in the middle of the slide at a relatively unhindered substrate, that would be relatively easy for this type of substrate to undergo an Sn2 reaction. The nucleophile is not impeded in any way, so it interacts with the sigma star orbital. We can get addition of the nucleophile, also the leaving group in an Sn2 reaction quite readily. On the other hand, in the green arrow here indicates mechanistically what has to happen for an Sn1. It would be simply cleavage of the carbon leaving group bond. That's not going to happen because primary carbocations, as it states here, are hideously unstable and not viable intermediates in any reaction. At the bottom of the slide, we have kind of a generic structure for a tertiary substrate, the other end of the spectrum. The nucleophile is impeded in its approach, and so we do not find Sn2 reactions on tertiary substrates. By contrast, Sn1 can happen quite readily under ionizing conditions. The carbon leaving group bond can fragment, and we can form a relatively stable tertiary carbocation.